50 casalènes de pétri tubes-bactériaux ou des chairs versus ce qu'on utilise pour les poubelles à paillasse. Donc en rouge, c'est l'impact du plastique exermique et en bleu, c'est l'impact du contenant vert équivalent. Donc vous voyez que chaque fois, cette phase de commentaire, l'impact du plastique est très supérieur à celui du contenant en verre. Si on regarde un petit peu la répartition de l'impact de chacun de ces matériaux, donc pour le plastique, en fait, ce qui impacte le plus, c'est la partie fabrication et le traitement de l'incinération et transport en fin de vie. Donc, quelques petites recommandations pour réduire l'impact plastique. Quand on veut réduire les volumes, c'est-à-dire si on a besoin d'un tube de ml, on ne va pas utiliser un tube 15 ml, on va vraiment garder un tube plus petit. Remplacer à chaque fois qu'on peut par le verre, mais sans condition qu'il n'y ait pas de conséquences, pas de problème d'hygiène et sécurité. Et réutiliser, c'est-à-dire si vous ne pouvez pas remplacer par le verre, quelquefois vous pouvez peut-être réutiliser plusieurs fois le matériau en plastique. Je vous remercie pour votre attention et peut-être un bon séminaire.
Bonjour à toutes et à tous, merci d'être à ce séminaire présentiel et online. Donc, je vais vous présenter Tania qui fait son doctorat, elle est en début de troisième année dans son doctorat à l'université Andrés Bello au Chili et que nous avons comme étudiante en mobilité avec Séminaire. Tania travaille sur notre terrain au Chili, sur l'AMR, sur les goélands, avec un système très similaire à celui qu'on peut voir en Camargue, sauf qu'il y a aussi des pélicans et des pingouins. Et c'est une vétérinaire de faune sauvage très spécialisée et très bosseuse sur toutes les questions de conservation, d'impact avec les communautés, qu'on a ramené pendant son doctorat à travailler un peu plus sur l'AMR. Donc, elle a fait beaucoup de terrain au Chili pendant 2 ou 3 années. Elle a fait beaucoup de labos au Chili aussi, et maintenant elle est chez nous pour travailler un peu sur la partie analyse de données bioinformatiques. Son doctorat qui se fait en collaboration avec l'Université de Chile, MIVEGEC et l'Université de La Rioja, dans sa co-directrice Carmen Torres. Donc, à toi, Tania, qui a dû comprendre la moitié de ce que je dis.
Bonsoir. Okay, so I'm going to do my talk in English. I separated my presentation in 5 main points. First, I'm going to talk about seabirds as sentinels of ESBL-E coli. Then, prevalence of AMR, or antimicrobial resistance, influenced by host and species. And then, diversity of genes and plasmids involved in AMR carried by seabirds, results, and final conclusion. So, what we need to know about antimicrobial resistance. So, actually, antimicrobial resistance is considered a global threat worldwide due to the misuse and abuse of antibiotics in human medicine and veterinary medicine. Particularly, these bacteria get resistance to antibiotics because of many human factors and drivers. And by result, we have many enzymes that bacteria acquire or produce because of this. The main ones that I'm studying is ESBL, that is a spectrum, amplified spectrum of beta-lactamase producing enzymes and carbamidamase producing enterobacteria. These ones are widely spread worldwide, and particularly in seabirds, it's reported that 80 wild-bird species are carriers of ESBL-E coli. So what we can say now is that E. coli or Escherichia coli is considered a sensitive indicator of AMR surveillance. And this is a map where we can see all the publications that have been done concerning ESBL in gulls. And what we got is that Europe, and particularly North America, there are many publications, but in South America, we have very few. In Chile, where this study is done, it's incipient and it's mainly done in very few species of seabirds or wildlife. It's very incipient. What is happening exactly in Latin America? So we have that these resistant bacteria are present in terrestrial environments and also in aquatic environments. They get in contact with many fauna, particularly in terrestrial environments. They can get in contact with companion animals, and in aquatic, they get in contact with seafood and seabirds. This seafood is eaten by the gulls, or the gulls itself drink the water, the salt water of the aquatic environment, or because of their behavior, they can get in contact with human waste. So they are in the landfills, or they can get in contact with wastewater, and also because of other kind of behaviors that they have, they can get in contact with other marine species. In this case, we can see this, that the gulls are very close to this pelican, and by this, the whole AMR antimicrobial resistance can happen. And additionally, they have migratory routes. So particularly in summer, many seabirds travel from North America to South America. So this can be a route also for AMR spread. What is happening particularly in Chile? In Chile, we have the Humboldt current large marine ecosystems. This ecosystem is very particular because they have a climate variation. So this make all the ocean very rich, and we have a very good fisheries. But as well, because of this, it is home of more than 50 seabird species. And we have in this study the pelican or Peruvian pelican, that is an endemic species of the Humboldt current, a resident species that is the gull, Larus dominicanus, and the franklin gull. This franklin gull is a migratory bird that visits us in summer. Summer for us in Chile is from December to March. So apart from the climate variation, these seabirds are not the same, so they forage differently. So they have different diet, and also they have different behavior because we found them in different environments. Maybe they are in wetlands, or they are only in the islands. Other ones are only oceanic. So all these traits could impact in the prevalence of ESBL-E. coli. So we are guessing that this also can impact the prevalence of the antimicrobial resistance. Additionally, we have that because of they visit us, the majority of them in summer, summer have their own characteristics. It is hot. We expect many people coming to the beach on holidays. They come with their companion animals. Also, the birds have different diet. They reproduce differently. Many of them are in breeding season, and some of them, they are not in breeding season, but they are gaining weight for that. They are preparing for that. And also, we expect less rainfall. But what happened to this? Because all of these and all of these characteristics, the only data that we have available right now is that in 2013, it is reported that between 30 and 54 prevalence of ESBL-E. coli in the franklin gulls is reported, only in summer though. So this is the only data we have. Apart from that, how this antimicrobial resistance is getting spread or disseminated. We have that in gulls, 70% of the studies involve antimicrobial resistant genes, and 34% involve mobile genetic elements. These mobile genetic elements are the ones that disperse these genes. So this is the bacteria, and this is the mobile genetic element. In this case, we are seeing a plasmid. And the plasmid is actually is one of the mobile genetic elements that is well reported and is considered widespread. So when the bacteria reproduce or just get resistant, the plasmid that contains the resistant gene transfer this to the other bacteria, and that is why the bacteria get resistance to the antibiotic, depending on the enzyme they carry. So what is the mechanism that produce this is called horizontal gene transfer. What we have right now in Latin America concerning the ESBL genes is the CTXM group. We have group 1, group 3, group 14, group 15, only in South America. And the carbapenems for America, particularly in Alaska, United States, we have the OXA48 and KPC2. However, there is no information in Latin America. The only thing that we have concerning mobile genetic elements is here in this study, but in Czech Republic, that is reported 11% of prevalence of integrons, another kind of mobile genetic element. And concerning the bacteria, the resistant bacteria is the E. coli in 30% of prevalence. But this E. coli was carrying great diversity of resistant genes. So what we can highlight here is that identifying the diversity of mobile genetic elements carrying many antimicrobial resistant genes in different seabird species could contribute to identify the molecular mechanisms of dissemination in a natural environment. So our hypotheses are the prevalence of ESBL-E. coli isolated from the gut of seabirds will be influenced by hot species and season. And second, the diversity of antimicrobial resistant genes and plasmids in seabirds will be influenced by trophic breeding. So our objectives are to estimate and compare the prevalence of ESBL producing E. coli isolated from species in Gulf, and in Gulf we are studying the catgut, Larus dominicanus, and the Franklin's Gulf, Larus pipixcan, and the Peruvian pelican, the Pelecanus thagus, between summer and winter, only for the catgut, because the other ones, the pelicans doesn't breed in winter, and the Larus pipixcans we couldn't, is not found in winter because it's a migratory bird. To identify genes and plasmid also involved in antimicrobial resistance in these 3 seabird species. So where is my study carried? Particularly in central Chile. In central Chile, we have the Valparaíso region, and inside Valparaíso region is Algarrobo community. In Algarrobo community, we have, this is the view of Algarrobo. It is a long coastline. This is a very popular destiny of many people to get on holidays, and they do a lot of sportive, aquatic sports, and also we can see a lot of marine fauna, and also it has an island, a puro guano island that many seabirds breed, and also it has many wetlands, so it is an ideal point to get our samples. What do we do, or what do we do? From 2020 and 2023, we collected 484 fecal swabs of seabirds from Algarrobo in summer of the 3 species, specifically from the catgut 249, Larus pipixcan 116, and the pelican 119, and only in winter 200 catguts. So all these fecal swabs were treated in Macomb agar, supplemented with cefotaxime and imipenem, 30 Celsius degrees by 44 hours. Then the lactopositive colonies that indicate as enterobacteria were purified to get these colonies and then identify them through molecular. Because we were searching Escherichia coli, once that we were sure that we were working with Escherichia coli, we do this double method to make us sure that it was a producer of ESBL. And then we do the analytical data using GLM. Concerning the carbapenem producing Escherichia coli, we extract the DNA, we send them to an enterprise called Microbes to do the whole genome sequencing, and we do the informatics. And we use these tools to get to know the antimicrobial resistant genes, multi-locus sequence types, plasmid types, virulence genes, mobile genetic elements, and do a phylogeny tree. And also, once we got all the sequence, we do a genome and plasmid visualization through DNA Explorer and Brief. These are the results. So in total, we have 263 confirmed ESBL-E coli. This is the specific result for the 3 species in summer and only for the catbird in winter. This is the prevalence. So we can see this comparison between summer and winter for the catbird. We see that the prevalence is almost equal and it has no significant difference. But we found a significant difference comparing these 3 species. And when we compared the year, we found that it was a significant, statistically significant difference for year 2022, with an estimate minus 1.8849. And we see that this effect is less or negative, sorry, and indicates that the prevalence for this year is lower than the other ones. And concerning the carbapenems E. coli, we confirm that there were 11, and they only were found in the catbird. So nothing for the other seabird species. These are the results, actually very low. We didn't find any difference between summer and winter. And this is the results specifically for the 4 carbapenems E. coli isolates. When we do the multi-locus sequence types, we found 2 different strains, 3 for 345 and 1 for 224. These are the resistant genes. It's not all of them, but the majority are the main ones. And we found the NDM1, that is very important for carbapenems, CTXM14, TEM1AB, TATA, and SUL1. The plasmids that we have here, the majority share the plasmids. Virulence genes, mobile genetic elements that we have here, insertion sequences, the majority of them, and all of them are present in the 4 isolates. The phylogeny tree, and we see here that 2 of them are very close than the other ones, but they are very related. When we have these strains and do the genome visualization, we can see here the gene in case of the NDM, New Delhi metallo-beta-lactamase. And when we extract this of the plasmid, we obtain that this is the plasmid, the circular plasmid that is carrying this resistant gene. And we do the comparison with all the 4 isolates. So it is here, it is reported the 4 isolates with the reference, and they all carry the NDM, the same NDM. So our conclusions is, sorry, what? our conclusions are that the prevalence of ESBL-E coli remain high, particularly in the Cold War and the Peruvian pelican during our 3-year period, and particularly in winter, only in the Cold War, of course, the prevalence remains high. So this would suggest that the AMR cycle is independent of season and temporality. So our hypothesis, the first hypothesis, is rejected because this prevalence doesn't have any difference between the seasons. And particularly concerning the genes, we see that many of them are shared between these isolates and in the same species. So it could be a suggestion that it's a possible mechanism. This horizontal gene transfer is happening between the species, the kelp. So it is a possible mechanism of AMR dissemination. So what is our next step? So now that we see that the gulls have these resistant genes, the prevalence is independent and remains high the whole year. Where is the source? So can be happening through the water. So in Algarrobo, they have many wastewater tubes that ends in the ocean. And we also take some samples there because of there are many dogs, feral dogs, companion dogs as well, and the fishes. So this is the next step to know the source of this antimicrobial resistant contamination. So thank you for the opportunity and for your time. And also to make the acknowledgement of Séminaire scholarship and Université de Montpellier.
Very much for your talk. Is there any question in the room? I was wondering, thank you very much. It was very nice talk and nice work. The 4 strains that are carbapenemates, maybe you mentioned it, but I didn't check. Are they from the same sequence type or not at all?
Yes, they are. 3 are from 345 and 1 is 224.
Okay.
But I specify the samples.
Interesting. And for the plasmid carrying the Bla-endian one, do you think they have exactly the same structure? Are they the same size, all these 4 plasmids, or is it completely different?
I think 3 of them are very similar, but the one that is of orange, is very different. It's short.
And this one is from the strain that is of different ST or not at all?
Good question. I don't think we have to.
No, it's the 59, and the 59 is this one. Yes.
Yeah, okay.
Tania, can you tell us what you found on the dogs and fish? Because you didn't say that we have the results, yeah.
Yes. What we already found in fish, we have nothing. Nothing concerning Escherichia coli, but we found Escherichia coli in wastewater and also in dogs. And specifically in dogs, we did a separation between feral dogs and companion dogs. So we already have data from that. So there are many dogs that are companion that holds ESBL-E coli, and many of them also, the feral dogs, hold them. So the prevalence of the dogs is approximately 10%, and the wastewater, I think, is 6%.
There's a question in the chat. Karen McCoy is thanking you for the talk and asks, How frequent are the carbapenem types in species in the region other than seabirds?
We look for the carbapenem in the dogs, and we didn't find any of them. We only isolated from birds and wastewater.
And I would just add that for other wild mammals that we have done in Chile, we have found none either. Wild foxes, mice, rabbit, and bats. No carbapenemas.
And the memory.
Yeah, and the wemul, yeah, the roe deer also is clean.
I have a question. Thank you for your talk. It's very interesting. It's very good. And in Chile, if I'm not wrong, I think among humans, NDM1 and VIM1, it's the most common among the patients with carbapenemas. Have you considered to looking in the prevalence in humans too, since you thought you talked it? I don't know what to say, but in the summer there is a lot of tourism in the area. So have you been considered in look for in humans?
What I considered as a human waste was the wastewater, but taking samples from humans, for example, in clinical settings, I didn't consider.
Not taking samples, but looking for in other studies that have conducted studies of prevalence to compare. Because if I'm not wrong, NDM is common in Chile and in human patients.
Mhm.
So it should be a good thing to add in the discussion section. I don't know.
Great, thank you.
And Karen on the chat asked what about seabirds outside Chile?
Well, that's a good question. Actually, what is carried in Chile, apart from these studies, is being carried out in the very south of Chile and in Argentina, in the Patagonia. And I think well, in Brazil, and
Only carba in Argentina. That's the only other place, and Alaska.
yes, but no, in Argentina they also done ESBL.
Yeah, but they have carba.
61, yes, 61% in Argentina from Kelly. Yes, and also the species that is more common to be done is the ghost. There is no information concerning the Peruvian pelican, for example. The only prevalence that I found was a very opportunistic prevalence in the United States, 40%, 14%.
And Philippe Agnew asked on the chat, could there be resistant genes circulating in bacteria other than those you select during your screening protocol?
We only look for enterobacteria. I don't know if I can say about the other bacteria that we found. No?
Yeah, we found one Enterobacter.
Yeah, we found Enterobacter, we also found Pseudomonas, Iomonas, and one Vibrio.
And there is, just to complement Philip, there is Salmonella circulating with AMR in birds. That's the only other study that we know.
The seabirds, they develop the disease or they are only the vectors for the bacteria?
We don't know the clinical affection that they may be carrying, but we know that they are the vectors and they are the chars that disseminate the antimicrobial resistance. But what this antimicrobial resistance does on the individual is not researched, but it's a very interesting question that I would like to address. But I don't know if I can talk about the report.
Yeah, of course.
But because then a parallel research that I going to carry out next year is about because we have avian influenza in South America, and many pelicans die among that. And we know that maybe this doesn't actually affect negatively in the pelican, but could be a driving factor to alterate the immune system. So that my intention is to report or to specifically work on the pelican and to see if there is a connection between this immune system and then human aggregation. So I'm going to compare different populations of Peruvian pelicans along the coast of Chile.
Yes, I have one question. Maybe it's stupid, but it's completely new for me. Yeah, I have some doubts with respect with the ecology of the seabirds with respect with your results. I am wondering myself how, for example, the phenology or I don't know, the maternal care can affect your results. Because yeah, it's very clear that the phenology in the seabirds is changing across the year.
I don't have specific results between, for example, pigeons, no, chicks and adults. But what I do know is that there are very few research that has actually done antimicrobial resistant research in chicks. And what they have discovered is that they have actually high prevalence. But it is not, it is only a report. They doesn't compare between that's range, stage, age range, and the adults. So it's undeveloped.
I have a question about the seasonality that you talked about. I was wondering, why do you conclude that there's no seasonality when on your graph you see that in winter the prevalence is really high?
Oh yes, it's high, but the difference is 8% difference. So when we did the ELM, we didn't see any significant difference. I think the p-value was superior than 0.05.
It's high in winter and summer. It's around 50% in both.
But both are actually very high, because what we expect in wildlife is that they remain actually very low, like 10%, 5%. This is for more than 30.
Tania, another thing that just to add is that at least in the first years that I was conducting the lab, we don't test it, or we didn't select the double disc, the negative double disc tests. So the prevalence of ESBL could be even higher, because some of the E. coli that grow in the medium, if they have the UMC gene along with the ESBL, they hide. I don't remember the word, but they mask the results of the disc. So and UMC is also very prevalent in animals in general. So you could have a higher prevalence if you did the good did the other tests to see if we have the UMC gene, test cefamicins to see if they are resistant to cefamicin, to see if the UMC could be in the gene, could be in the strain. So your prevalence should be even higher, I think. So this is also
Do you mean for the ones that are not reported as E. coli?
Yeah, no.
Or for
the
ones that are,
because you did the whole genome sequence, for the positive in the double disc method, right? If you have an E. coli that has UMC along with the ESBL, the test would be negative.
Yes.
You don't test it, so could be ESBL-2.
Yes.
In the
Uh-huh.
in your negative could be a false negative. So your prevalence should be higher in this case.
Well, no, because we had Carmen asked us to add antibiotics for that. Yeah.
This is another confirmation. I did it in Chile, and then in Spain, they did it again.
Yeah,
So
but
we test the
it's
MC.
the
We test the MC.
okay. I didn't know that. And did you find some with MC?
Yes, but pretty low. I don't remember the number. I didn't put it here, so.
I think it's less than 3% or something. Yeah.
Yeah, very few. Like I think 2.
It's great to put these two because it's common to see high MC in animals. I don't know the last publication, but it's very common MC in animals in general.
And we also found the Escherichia marmorae. Yes.
But we are sequencing.
Yes.
Yes. Thank you very much for your presentation. I have a just quick question. Maybe I skipped the detail, but did you make any phylogeny of your strains, the sequence? And
Of the carbapenemas.
yes, of the carbapenemas or the other one that you found with the other one that we found in the genomic library around the world to see where it is closer.
Yes, I'm doing that this week.
Okay, so that's fine.
And Tania could mention that she did that on Carva, but she has 100 samples ongoing for genomics on ESBL. So all the ESBL for each species, they have been sent to a microNG and are on progress. So she did it for Carva, but she has a lot of data.
Yes.
Any other questions? Thank you very much for your talk and your answer.